Hello students. Today we will talk about another very important concept on pollination. So we have learnt about the male and the female gametes, right? So we have learnt about the male gametophyte and female gametophyte, and what you have learned is both the male and female gamete, right, that is going to be produced by the male and female gametophytes, they are going to be non-motile, right? So what do you mean by non-motile? Non-motile means they cannot move, right? So the obvious question is, as the male and female gametes are produced in two different structures, that is male and female gametophyte, how do they reach each other? Because for the next process to go on, for the fertilization to occur, both the male and the female gamete must reach, or they should be together. And being non-motile, that raises a question on how will they come together, right? So the process by which the male and female gametes are coming together, in other words, what actually happens is the male gamete reaching the female gamete, that is what is called as pollination. So the pollination refers to the transfer of pollen grains from the anther to the stigma of a pistil, and that is going to be called the pollination, right? And this process is going to bring the male and the female gametes together. So what you have to keep in mind is, for example, you have the anther, right? And this anther is going to release the pollen grains, right? So the pollen grains are going to be released when? Not always. The pollen grains are going to be released by dehiscence. So what do you mean by dehiscence? Dehiscence is the release of the pollen grains by the anther when the pollen grains are going to mature. So upon maturation, the anther is going to dehisce and it is going to release the pollen grain. So once we have the pollen grains, right, they're not going to stay there. They need to move. Although they are non-motile, they need to be moved with certain help of agents, right? They are called pollinating agents. Now this pollinating agents, what they will do, see, pollinating agents here, you have to keep in mind they can be biotic or they can be abiotic, right? So we'll talk about that in future video, but in simple, what you have to keep in mind is biotic means the living organisms, and abiotic means non-living components, for example the wind, the water, and they are going to act as agents of pollination. So for the pollination to occur, this pollinating agents, they are going to take the pollens, or they're going to allow the pollens to reach the stigma, right? Stigma of what? Stigma of pistil. And when we have the pollen grain reaching the stigma of pistil, that basically completes the process of pollination. So in simple term, the pollination is going to allow the transfer of pollen grains from the anther to the stigma. Now why it is important? Because you know that the stigma is going to lead to the ovary, and in the ovary we have the female gamete, that is the egg cell, right? Stigma, style and ovary together, they're going to form the carpel, and that is why the pollen grain from the anther must reach the stigma to continue the process of development in the sexual reproduction in the plants, that is to form the single cell structure called the zygote. Now depending on the source of pollination, right, we can divide, or the types of pollination can be of three different types based on what, right? Types of pollination based on source of pollen. So based on source of pollen, what we can do, we can divide all of this into three different types. So types of pollination based on source of pollen is three types. So obvious question is, what are the three types? So the three types are, first is going to be called autogamy, second is called Geitonogamy, and the third one is going to be called the Xenogamy. So we're having these three different types of pollination, and this different type of pollination is based on the source of pollen. Now I'm sure you have all heard of something called self and cross pollination. Okay, so these are nothing but the self and cross pollination, but we're going to learn in terms of these three different terms. So the first thing that you have to keep in mind when we talk about autogamy is the involvement of only one flower. Okay. Now, as you can see in the image, now this particular part is the stigma. So you can see here, these are our male, or they contain the male gamete that is going to release the pollen grain. And when they're going to release the pollen grain, these pollen grains are going to come and settle on the stigma, this particular part. Now when this process, the pollen grains are going to come from the same flower and reaching the stigma, we are going to call that as autogamy, or in simple you would have heard about the self pollination process. Now the obvious question is, in what condition is it possible? Is it possible in all the condition? No. So one of the condition is there should be synchrony between the pollen release or pollen maturation or release, we can say both, pollen maturation or release, and stigma receptivity. Now what do you mean by this? So at a single time, at the same time, both the pollen grain should be released and the stigma also should be receptive, that is, it should be mature enough to receive the pollen grain and continue the developmental process. Now if there is a difference at the timing, if let's say the pollen grain is released much before the stigma is receptive, what is going to happen? The stigma is going to receive the pollen grains, but probably it is not going to retain it. It is not going to continue the development. A synchrony is very important between the pollen maturation or pollen release and the stigma receptivity. Now the second important point that you have to keep in mind, or that is essential for the autogamy, is going to be the close position, okay, or close arrangement of stigma and anther. So here both the stigma and the anther must be close to each other, so that whenever the anther is going to release the pollen grain, they're going to fall directly on the stigma. So for the autogamy, the plant must have these two features so that they will have a complete pollination within the same flower. Now let us talk about three different examples. Okay, now why are these examples important? Because they are unique in the sense that they're going to produce two different types of flowers. What are these? We'll talk about in a minute. So one of the example is going to be Camelina. Okay, then we have Viola, and then we are going to have the third plant, that is going to be Oxalis. Okay, so here these plants produce two different types of flower. And what are these two different type of flowers? Now these are called, one called Clistogamous and the other one is going to be called the Chasmogamous. So these are the two different type of flowers that these plants produce, right? Camelina, Viola and Oxalis. Now what it has to do with the concept that we are learning, autogamy? Let us see that in this particular image. So as you can see here, this particular plant is trying to show these two different type of flowers. One is going to be called the Chasmogamous flower and second the Clistogamous flower. Now what you have to keep in mind is the Chasmogamous flower is like the regular flower. Okay, these are like the regular flower. What do you mean by regular flower? A regular flower with well exposed stigma and anthers, as you can see in this. So this is an example of the Viola plant, and in the Viola we are talking about the Chasmogamous flower. So these are regular flower with the anther and the stigma which are well exposed. Now the second category is the Clistogamous flowers. Now what do you see in Clistogamous flowers? So here there is something you have to keep in mind, that these flowers are going to be underground, right? So these are going to be underground layer, and you can see these are present inside around the roots, as you can see. Now these flowers are going to be underground and they will never open. So these flowers, they never open, they remain closed throughout. So what is the benefit of this? Because they are Clistogamous flowers, because they never open, they do not need, right? So there is no need for pollinating agents. Okay, there is no need of pollinating agents. And what is the benefit of this? The benefit here is there is going to be an assured set of seed, right? Assured seed set. That means whether you talk about the pollinating agents being present or absent, there is guaranteed that there is going to be a seed formation in this case, in case of Clistogamous flower, because remember the flower is not opening, the flower is going to remain always closed. There is stigma, right? There is anther, there is going to be release of pollen grain, the pollen grain is going to fall on the stigma and the entire process is going to go on, but without the flower ever opening. So there is no need of the pollinating agent in case the flower is Clistogamous flower. And as you can see, this is an example. Again, this is from viola. So this is the viola plant's Clistogamous flower. So this flower, as you can see, they will never open. They're underground, they will never open, but they will give a assured seed set. So in this case, that is, for example, we are talking about Clistogamous flower. In this cases of Clistogamous flower, is there any drawback? So here one of the drawback is going to be there is going to be less variation. Okay, and that might even lead to something similar to that is called inbreeding depression. So the variations here are going to decline because there is no different type of pollen grains. It is the pollen grain from the same plant which are genetically similar. So you have to understand that variations are going to enable the plant to be more stable, the population to be more stable. So the plants or the organism always try to accumulate variations, but in this case what happens, variations are going to be lesser, and that is what a drawback of the Clistogamous flower. So that is what you have to understand with autogamy, Clistogamous flower and Chasmogamous flower. So to summarize, Chasmogamous flowers are regular flowers with well exposed stigma and anthers, whereas Clistogamous flowers, they will never open, they will always remain closed, they don't require a pollinating agent, but they will give rise to associate set. But again, there is going to be presence or development of less variation, which might be a drawback. Now the second thing is going to be Geitonogamy, and the first thing that you have to keep in mind is going to be there is going to be involvement of two flowers, right? When we talk about two flowers, as you can see in both of these cases, so these two flowers, if you focus here, they're actually from the same plant. Okay, so here we are keeping a condition, there are two flowers involved, and these two flowers are from the same plant. In this case also, these two flowers are from the same plant. So here what is going to happen is there is a involvement of, let's say the stigma, this is stigma, so the anther from here is going to release the pollen grain and that might pollinate the stigma here. Similarly, here you can see these are the anthers, and these anther might release pollen grain that might pollinate this particular stigma. So here we are talking about the involvement of two flowers, right? What? From the same plant, right? Only one plant is going to be involved here. And if we try to understand, this refers both to cross-pollination and self-pollination or autogamy to an extent. Now, if you talk about functionality, so here functionally, right, they are going to be similar to that of cross-pollination, right? Why cross-pollination? Functionally, because they are involving two different flowers, so cross-pollination. But when we talk about genetically, right, whether are they different genetically, the answer is no, they are the same plant, so genetically they are the same. So genetically, if you see, Geitonogamy is nothing but the autogamy, right? So functionally they are similar to cross-pollination, but genetically they are similar to that of the autogamy. So Geitonogamy is pollination involving two different flowers, but they belonging to the same plant. Now when we talk about xenogamy, again here there is going to be involvement of two flowers, but the difference here with the previous case is these two flowers are from, right, different plants. These two are from different plants, but these two plants belong to the same species, right? So they are from the same species, but two different plants, two different flowers, and there is pollination between them. So you can see here, right, this is the anther that is going to have the pollen grain. Let me change the color to something else, and here they're going to release the pollen grains, and that is going to, here you can see this part is going to be the stigma. So this pollen grain is going to be released from this particular flower. This is from, let's say, plant 1, let's say, and that is going to reach the plant 2. So two different plants are involved. Similarly here, otherwise what we can have is this particular anther might release pollen grain from this particular plant, and this might reach the stigma of this particular flower from the plant 1. So here there is going to be involvement of two different flowers, but they are from two different plants. They are again from the same species. The species cannot be different. Again, in future we'll talk about pollen pistil interaction, and we'll talk about compatible and non-compatible pollen grains, right? So this is the only pollination type, right? We talked about three different types already. We talked the two, and this is the third one, that is autogamy, Geitonogamy and Xenogamy. Out of these three, only the xenogamy is going to bring genetically different pollen grains. So it is going to bring genetically different, right, pollen grains to the where? To the stigma. So dear students, this is what you have to understand with the pollination and the types of pollination, autogamy, Geitonogamy and xenogamy, with regards to the source of the pollen grains.